everyone, and welcome to our third webinar on managing nature's resources in organic production under water-limited conditions. Today we're going to be talking about legumes for nitrogen supply under water-limited conditions. I'm Karen Clausen from the Manitoba Organic Alliance, or MOA. We'd like to begin by acknowledging that we're in Treaty 1 territory, and the land is the traditional territory of the Anishinaabe, Cree, Oji-Cree, Dakota, and Dene peoples, and the homeland of the Métis Nation. In addition to hosting this series of webinars, we also host other events and are going to be at Ag Days this year, and we have our own conference, Prairie Organics Conference in February. So check out our website to find out more about this. Our partner in bringing this webinar series to you is the Natural Systems Agriculture Group at the University of Manitoba, and these webinars are also hosted by Dr. Martin St. We'd like to start out by thanking our sponsors for this, who've made this extension work possible. So first of all, we have FCC, Grain Millers, Manitoba Harvest, ProCert Organic, Theory Mesh. We're also funded generously through the Prairie Organic Development Fund, Manitoba Agriculture, and from the federal government through the Canadian Agricultural Partnership. If you're an agronomist, you'll get CCA credits for the webinar. At the end of the webinar, we'll put up a unique code that you can scan with your phone. Later today or tomorrow, you'll receive an email with a link to this recorded webinar as well. If you have any questions that occur to you during the chat, please put them into our question box, and Martin will get to those at the end of the presentation. I want to hand this over now to Dr. Martin St. from the University of Manitoba.
Okay, thank you very much, Karen. Good afternoon, everyone. I have the pleasure today of hosting our guest, Dr. Mervin St. Luce, and we'll hear from him in a minute, but let me ask him to please turn on his camera so we can all see him, and then I will give the introduction. So Dr. Mervin St. Luce is a research scientist in soil fertility and cropping systems at the Swift Current Research and Development Center of Agriculture and Agri-Food Canada. He's got a broad knowledge of legumes in not only temperate farming systems, but also tropical systems. He received his PhD from McGill University in 2013, and he has been in Saskatchewan working on his soil fertility program for a number of years. His current research involves the use of legumes to enhance nitrogen use efficiency, carbon sequestration, soil health, and biodiversity in a number of different sort of climatic conditions. Now, I first met Dr. St. Luce in Chicago, actually, at a meeting of people who were running long-term experiments. And so Mervin is looking after, he leads several long-term studies at the Swift Current Agriculture and Agri-Food Canada Center. And so we have that mutual interest in what is the sort of long-term effect of management on things like nitrogen supply, on water conservation. And so with that introduction, I've actually wanting to just ask our guest a couple of little informal questions before we get started. So first of all, hello, Mervin, and welcome.
Thank you, Martin, and thank you for the invitation. Thank you all for attending this webinar. It's a pleasure to be here.
Great. It's good to see you again. I look forward to when I can see you in person again.
Same here.
Yeah. So Mervin, I'm going to ask you a few little questions just to warm you up. Now, I see from the Agriculture and Agri-Food Swift Current website that your research interests include resilient cropping systems and the development of best management practice to improve soil health, nutrient use efficiency, and particularly nitrogen. So I guess my question is, what got you interested in these subjects in the first place?
Yeah, and that's a good question, and I would like to start by saying, I mean, in agriculture we have major challenges And one of the biggest challenges is to feed a growing population. But also to do that in a sustainable manner, that we do not, we reduce our impact on the environment. We do have climate change to consider as well. I mean, look at this year with significant drought across the prairies. So how can we produce food sustainably that is also profitable for the farmers within this context of climate change, nutrient use efficiency, greenhouse gas mitigation. Now we're talking about net zero production. So my interest in resilient cropping systems basically is to be able to develop systems that can withstand and bounce back sort of to shocks, whether they be biotic or abiotic shocks. And we need to be able to do that to take advantage as well of good growing conditions. So having different aspects of research in terms of disciplines, so not just individuals in soils, but we have in agronomy, microbiology, soil microbiology and ecology, economists, to be able to look at production systems that can be productive, efficient, resilient, and basically achieve the goals that we all want to be able to achieve to enhance system. So we're not looking at a crop, we are looking at an entire system. So it's not focused on wheat or canola, it's a full system. So we're looking at a systems approach and developing a resilient cropping.
Okay, thanks for that. So what I think we all, there's parts of our jobs that we really like and parts of it that are more like work. What aspects of the research do you find the most interesting? I mean, clearly there's a mission, there's a mission about food security, so I think that's really gratifying and also challenging. But of your day-to-day work, what do you find most interesting?
Research itself is exciting for me, and that's why I got into it. So getting to discover, to innovate, and certainly to work with other individuals. I really like collaborating with people like yourself, Martin. And
okay. Thank you.
To be able to come together and to try to solve a lot of the challenges. It does not take one individual to do that. We have complex challenges, bringing different heads together. That is exciting for me, working with a diverse group of people from right across the country as well as internationally, and just personally focusing on the things that I can control. So making sure that I do my part within the bigger picture. I'm not here to save the world, but at least I can do something that I can make some improvement in the area that I am at this time. So research for me has always been, especially science, always been of significant interest, and I have not regretted one day getting into soils and soil fertility.
Fantastic. So you mentioned that you have a sort of a team approach. You mentioned that, different disciplines. You also at Swift Current have some initiatives in organic production. I think Miriam Fernandez started that a number of years ago. Is that organic program part of sort of the team effort at Swift Current, or how is it organized these days?
I would say it's the organic production or the organic research program in Swift Current is mostly Miriam. So when I arrived, she had a program for a number of years ongoing, and she invited me to collaborate with her. So I'm contributing in some ways, especially looking at nitrogen cycling and the organic systems, and also organic matter as well. So that's mostly my my contribution or my role with Miriam, and I do appreciate the fact that she invited me to work with her and some of the farmers or the group that she has put together. But that's a small, I don't want to say small, but the bigger part of my program is conventional. So I'm most of my days spent in conventional research area, not in organic. But I do enjoy working in organic systems. I mean, I grew up in the Caribbean, and we do have a lot of organic farming. Basically, we do not use a lot of chemical inputs. So I have grown up accustomed to that.
Yeah, yeah. The well, it's so interesting how many agronomists dabble in organic also, because they're curious about what of those practices could be used in other types of agriculture. So
correct.
Thank you for joining Miriam and spending a little bit of your time working on organic production.
Yep, for sure.
Yeah, so let's shift our thinking to drought, and we're just about ready to have you start your seminar, but I wanted to maybe end off with a little question about, what experiences from working in other places in the world, maybe different than the dryland prairies, have informed your work on legumes and cropping systems? Because, I mean, we know that legumes can be included in cropping systems all over the world, and they are. So what experiences from other places have informed your work, maybe the work that you're doing in Swift Current now?
Yeah, I would say most of the experience I had was while I was in Quebec, actually. In the Caribbean, we do not grow pulses or legumes widely in terms of mass production. Typically, it's mostly subsistence farming. So especially like pigeon peas, that's one of the more popular, which is like a shrub, a small shrubby plant. But in Ontario, Quebec, I mean, it's mostly corn soybean rotation. Sorry. So my work there kind of helped me to understand a little bit better in terms of how legumes interact with other crops, in terms of nitrogen supply, breakup of pest and disease cycles, and all of the other benefits that legumes provide. Interestingly, while I was in Quebec, I worked on doing my PhD and my postdoc. I worked on a research project that was taking place in Western Canada, but I was in Quebec. So I did have sort of an external or virtual working relationship with a lot of the researchers in Western Canada. And some of that I will present some of that results today. So it's quite a drastic difference between Eastern Canada, where it's much more humid, versus the prairies. You can see the impact. So soybeans are not performing well here because of the drier conditions, and they do well in the more humid areas. And here we have a lot of lentils and field pea, and much bigger farms with cereals and oilseeds, especially canola. So the drought has a big impact in terms of not just the performance of the cereal, but of the legumes themselves, and how much they can fix nitrogen, what is the potential for supplying nitrogen to the next crop, because it depends to a great extent on mineralization. And without moisture, you do not have that turnover going to take place. So we can have good fixation, but have not the best response in the following year, because maybe during that year we have drought, like this year. So all of that nitrogen is tied up in the soil.
Oh,
so
that's a perfect segue into your seminar, and I think that's one of the questions that often comes up is I've got this green manure, I've got this legume, but it's so dry. Am I getting any of the nitrogen? So I'm going to turn it over to you, Mervin, then, and thank you again for taking time to joining us. I'm going to turn my camera off and let you share your screen, and then we are all
Okay.
yours. And if there's any technical difficulty, just articulate it, and there is somebody in the background who can help you.
Okay, I'm gonna try to share my screen. Yes, thank you. You can let me know when you can see my presentation.
Yeah, now we see your presentation with the second slide over on the right. So
Mervin, you can go
Okay,
up
so
to
I have
display
to.
settings. No, if you want to go back there, sorry,
Okay.
and we'll do that swap again.
Okay, so I have to share screen again.
Yep. And then start your slideshow from beginning. Is it on? Okay, now up to display settings, top left, and it says display settings on your with your notes.
All right, right. Yes, I forgot that part.
And then swap. You bet. There you go, sir.
Yes, thanks for the reminder. Okay, so you can see that perfectly now.
Yes, it looks really good.
Okay. All right then. Thank you. Okay, so thanks everyone, and thanks for coming to this webinar. So today I'll be presenting on legumes for end supply under water limited conditions. So I'd like to start off by just showing some of the recent production data from Statistics Canada, showing the harvested area of pulses in the Canadian prairies. As you can see here, lentils and field pea dominate the landscape. We do have some increase in soybean, especially in the more eastern prairie regions, compared to the more drier areas for the west. We do have some chickpea as well, with dry bean and faba bean. However, we see clearly that field pea and lentil are the two dominant pulses that are grown in the Canadian prairies. So these contribute significantly towards farm production as well as the economy on a whole. So just to give a refresher in terms of how do legumes supply nitrogen, I'm sure we all know the mechanisms by which this is accomplished. So we have legumes with that can fix nitrogen from the atmosphere because of the relationship with bacteria in the roots. So these legumes, they take atmospheric nitrogen and convert it for their own use, as well as adding some of that to the soil. Now, this is not the only way that legumes supply N. They also supply N through the decomposition. So biological nitrogen fixation is bringing the nitrogen from the air to the plant and to the soil. But then the residues, once after the crop is harvested, can be mineralized and provide available N to subsequent crops. And that comes both from the above ground biomass as well as below ground. I just want to point out again, legumes do not only supply nitrogen, they also provide non-nitrogen benefits, such as soil moisture availability and breakup of pest and disease cycles, which I will briefly talk about later in this presentation. There are some factors that can definitely affect the potential of legumes to supply nitrogen. One is the legume species themselves. As you can see in this table, legumes have different capabilities or potential for fixing nitrogen. We see here that faba bean tends to have the highest potential for fixing N, and generally dry beans with the lowest potential for nitrogen fixation. as well the growth and performance of the legume, including green yield and biomass, the actual quantity of nitrogen that is fixed, pests and diseases that could affect the legumes themselves, and of course, site-specific conditions, including weather, soil texture, and other growing conditions. I really like this quote here from one of these articles from Peoples et al. From Australia, where they explain that the amount of N fixed by any legume is directly influenced by factors that affect growth and development. So whatever the conditions are during the growing season, this will have some impact on the ability of that legume to fix nitrogen. So I would just like to take some time to present some of the results that we have achieved so far in some studies that have been completed, as well as some current research programs that we are undertaking, as well as from some long-term experiments, which gives us a nice picture of dynamics and changes over time. So the first study I would like to talk about is a study that was conducted across Western Canada from 2009. This study was led by Cindy Grant. I'm sure most of you are familiar with Cynthia Grant, who retired from AFC. So there were two experiments conducted. The first experiment was conducted across these 7 sites: Beaver Lodge, Lacombe, Lethbridge, Swift Current, Scott, Indian Head, and Brandon. So encompassing the different soil zones across the prairies. Sorry. And then the second experiment was conducted at Lacombe, Lethbridge, Scott, Melfort, Indian Head, and Brandon. So giving us a nice picture of what would happen under these different growing conditions. Just to provide a description of the experiment, the design and treatments. So experiment 1 was conducted from 2009 to 2014, and experiment 2 from 2010 to 2012. So in both experiments, the first year we had wheat, canola, faba bean grown as for seed, also grown as green manure, field pea, and lentil. And then following that, we had either canola or spring wheat with 5 different nitrogen rates through a split plot design. And these crops were followed by malting barley or canola respectively, also fertilized. And then for experiment 1, we had canola under the 5 nitrogen rates. And an interesting thing about the experiment 1 was that in 2013 and 14, we stopped applying nitrogen. So in that case, we wanted to see whether or not the crops grown in 2009 still had any impact on yield and quality of spring wheat and canola. So some results here. So the first graph here shows the quantity of nitrogen that is returned in the biomass, so excluding the seed. We see here, as expected, much higher nitrogen returned for green manure, followed by the other legumes, so faba bean, field pea, and lentil, and then cereals, wheat, and oilseed canola. In red, you can see the quantity of N that is actually exported in the seed. So for the legumes, a significant amount of nitrogen is actually taken off the field in seed, which is the product that we want. But you have to consider that that is an export of nitrogen. And compared to the green manure, everything is actually returned to the field. So we did find higher nitrogen returned by legumes compared to canola and wheat. We also had a higher spring nitrate concentrations following the legumes, especially green manure and lentil. And we had low N in spring following wheat and canola. Now you will realize that faba bean also had a low spring N concentration, and that although fababin had the highest potential to fix N. So this is a prime example of how growing conditions can affect the ability of a legume to fix nitrogen. In that case, in most of the sites, fababin did not perform well. There were lots of diseases, and the yields were very low. So that affected the nitrogen that was returned as well as nitrogen that was fixed by fababin across these 13 site years. So in conclusion, we can say that the legume impact is dependent on the legume species and whether it was grown for seed or as green manure. This is some interesting results when we only consider the plots that did not receive nitrogen. That is the following year. So when canola was seeded in experiment 1, we see here that in Brandon there were no significant differences in canola yield among these preceding crops. However, at Lacombe we found a higher canola yield after green manure and fababin, and then we had lower yields following field pea, wheat, and canola. At Scott, we found a higher canola yield after green manure, and then lower yields after canola, fababin, field pea, and wheat. So the legume impact definitely is site-specific, and it depends on, again, on the legume species. And generally, we had higher yields after green manure across most of the sites, and that was due to a greater amounts of N-rich residues that was returned by the green manure crop. Now, as I mentioned earlier, we stopped applying nitrogen for 2 years, 4 years and 5 years after the legumes were grown, just to see whether we can still find an impact. However, we found that yield and quality response were due mostly to the previous N that was applied in 2010 and 2012, and not due to the crops grown 4 years prior. So the legumes had no impact actually 4 years later across most of the sites. But we did find some specific interesting results. So at Indian Head, we found spring wheat protein concentration was slightly higher where wheat or field pea were grown 4 years prior. At Beaver Lodge, we found greater protein concentration at the highest N rate where fababin, green manure, or lentils were grown. So that interaction between the N rates that were applied between 2010 and 2012 with the fababin, green manure, and lentil contributed to a higher protein concentration at Beaver Lodge. At Swift Current, it was interesting that we found protein concentration in the unfertilized plots was lower where wheat or canola rather than legumes. So the legumes had some impact on protein concentration 4 years after they were grown at Swift Current in terms of protein. Finally, the crops grown 4 years prior, they affected crop N accumulation. So generally, we found higher nitrogen uptake in plots where legumes were grown 4 years prior. 5 years later, when canola was seeded, we found no significant effect of crops grown in 2009. The yield and quality as well as nitrogen uptake by canola was due mostly to the previous N application. So that was N applied in 2010 to 2012. And of course, site-specific effects. So we found magnitude of the responses varied by site due to differences in organic matter content and soil texture, as well as growing conditions, especially moisture. So those were some very interesting results that we found. In experiment 2, I'm just showing here the average results across 6 sites. And this was wheat grown after these crops. So as you can see, firstly, wheat response on wheat stubble was lowest, followed by canola, and then the pulses. If you look at specifically in plots where nitrogen was not applied, you can see clearly that we had higher yields with no fertilizer application for pulses compared to canola and wheat, especially after green manure and lentil. Now, we estimated the economic nitrogen rate from this data set, and as well as the economic yield. So what we found for wheat grown on a wheat stubble, the economic nitrogen rate was 76 pounds of nitrogen per acre, and the economic yield was about 54 bushels per acre. So if we do this for each of the different stubbles, we do see here that compared to wheat, we found 16 to 18% more yield. So yield, economic yield was about 64 bushels per acre, but that required 27% less nitrogen after those legumes to achieve those yields. So more yield, but also less nitrogen required. So also across the sites, as you would imagine, these economic yields and N rates, they varied significantly. They were quite low at Lacombe because very high soil organic matter content, as well as good growing conditions in adequate moisture, compared to Brandon and Swift Current, where we had drier conditions, low organic matter content, and the responses were not, they were mostly linear in most cases compared to a quadratic response. So canola following wheat in these plots, we see more of a linear increase in yield on canola stubble, and similarly on wheat stubble, where we found more of a quadratic response. So it increased up to a maximum and then declined afterwards on legume stubble. Similarly, we do find where in plots that were not fertilized, we do find that canola yield was much lower on canola stubble compared to faba bean green manure and lentil stubble. So doing the same exercise where we determine the economic nitrogen rate and yield, similarly, we find that compared to wheat and canola, we did find similar yield, but this time 40 to 55% less N fertilizer was required on this legume stubble. In both cases, that is when wheat was grown and when canola was grown, we did find among the legumes, the response on faba bean stubble was lowest. So we did have lower yields on faba bean stubble, and also close or a little bit higher economic nitrogen rate on faba bean stubble, for some of the reasons that I explained earlier in terms of the performance of faba bean across those sites. So on canola, as I said, we have a linear increase on canola stubble, but on the other legume stubbles, we did have more of a quadratic response. So the legumes were able to provide nitrogen to both wheat and canola, and required less actual nitrogen fertilizer to achieve maximum yield. So I just wanted to explore a little bit further in terms of how does legumes affect the system. We do know that legumes provide nitrogen, but there's lots of research that suggests that they provide non-nitrogen benefits as well. So in this slide here, we can see 3 different responses across 3 sites. So if we start with Brandon, what we can see is on the y-axis, we have wheat grain yield, and on the x-axis, we have nitrogen uptake by the crop. So at Brandon, we can see as yield increases, crop N uptake also increases, but all of the points are along the line. That means nitrogen contributed to grain yield at Brandon. So at Brandon, this was mostly a nitrogen benefit. So that was the limiting factor. All of the points, including the green manure and lentil field pea, you do find them higher up on the curve. However, they increase as as wheat grain yield increased. However, if you look at legume, we can see that there are some points that for wheat specifically that at the lower end of the curve, and then we do find more points for the other stubbles higher up. You can see for the same nitrogen uptake, we have differences in grain yield. So for example, we have 180 kilograms of nitrogen uptake for a wheat stubble, but for that same uptake, we have a higher yield if the stubble was filthy. Similarly, we get similar results at Scott, where we do find for the same nitrogen uptake, we have differences in yield. So at those sites, we had both a nitrogen and a non-nitrogen benefit. So nitrogen was not only the limiting factor at Legume and Scott. As I mentioned earlier, Legume had a very high organic matter content compared to the other sites. So that was a classic example of where legumes can have an impact, not just only on nitrogen. So what are some of these non-nitrogen benefits? One that is specific, especially to the prairies and especially the semi-arid area, is moisture. Moisture controls all of the processes in our sites here. So pulses generally have most of their roots in the upper layers of the soil compared to cereals and oilseeds that go deeper. So that means the pulses will actually use less moisture, less water than cereals and oilseeds. So the crops that are following pulses typically have more moisture available to them than if they followed wheat or canola. So that's one of the non-nitrogen benefits that pulses provide, and this is very critical, especially in Western Canada. Now, specifically for growing pulses or legumes, we do need to estimate what is the nitrogen credit to be able to adjust our nitrogen rates for economic as well as environmental reasons. Over-applying nitrogen can reduce nitrogen use efficiency and also is more prone to be lost from to the environment. So we do need to target better nitrogen use efficiency. And one way to do that is to have a good estimation of the credits nitrogen credits that legumes provide. Now, there are two general ways of estimating nitrogen credits. One way is the traditional method, but this method assumes there is only a nitrogen benefit and there is no non-nitrogen benefit. So in that case, this is N rate applied to a non-legume or on a non-legume stubble that produces an equivalent yield to the unfertilized non-legume on a legume stubble. So if we take, for example here, wheat yield on canola stubble versus wheat yield on field pea stubble. So we can see here that for the 0N plot in the lentil stubble, produce this amount for the equivalent yield on canola stubble. What was the N rate that the canola stubble required to produce the equivalent yield when grown on the field pea stubble where no N was applied? So we see about 30 kilograms of N per hectare. So that is how the traditional method works. The second method is the difference method, but this method compares the differences in nitrogen, in economic nitrogen rates. So if we take the same stubble, so we have canola and field pea as our example. So if we look at the economic yield and nitrogen rate on canola stubble, and then we look at it on the field pea stubble, what we can see is that we have a higher yield on field pea stubble and lower nitrogen rate required compared to canola. So that difference is the credit that can be applied. However, this method tends to be more accurate, and compared to the traditional method, we typically overestimate the nitrogen credit. So using that approach, we can estimate nitrogen credit for the different legumes on the wheat stubble as well as canola stubble. So the traditional method, you can see here that the nitrogen credit was ranged from about 55 to 95 pounds per acre using the traditional method, but using the difference method, it's about 19 to 21 pounds per acre. On canola, compared to canola stubble, it ranged from 25 to 37 pounds per acre using the traditional method, compared to about 18 to 20 pounds per acre. So there is quite a huge gap between these two methods, but generally the difference method tends to be more accurate, and because it considers both the nitrogen benefit and the non-nitrogen benefit. So now I would just like to show some of the results from our long-term experiment in Swift Current. We have 3 long-term experiments. The oldest one, which is called the old rotation, actually, it was initiated in 1967. But the one I'm going to be talking about today is the new rotation, which was initiated in 1987 in a no-till system. And it was designed to compare with the old rotation, which is under conventional tillage. In this area, we have a silt loam soil with pH about 6.5 and organic carbon about 1.8%. There are several different cropping systems, but for the purpose of this presentation, I will be focusing on 4 of them. So there's the fallow wheat, green manure wheat, continuous wheat, and our diversified system, which started in 2003. So wheat, canola, wheat, field pea. All of these crops were fertilized except the green manure. Sorry, and the peas. And the nitrogen recommendations for wheat was soil nitrate to 24 inches plus fertilizer to provide 80 pounds per acre on fallow and 65 pounds per acre on stubble. So I did say that the the experiment is no-till, but the green manure phase is actually not a no-till system, and that has been since its inception. The green manure is terminated by using cultivation, more of a shallow cultivation. We have used either a V-blade cultivator or tandem disc plow. And in fall, this is what the green manure plot looks like. It is not, so in that case, the green manure phase is not no-till, and we intend on changing that next year to herbicide termination. Previously, the green manure was terminated at full bloom, but some of the results show that it was using a lot of moisture, so changed to termination at 20% flower since 1993. So we can see over time we have a huge dynamics in terms of growing season conditions. So that gives us an ideal opportunity to look at changes as we go along. We had some drought in 2006, 2007, and then we had also above normal precipitation in 2010 and 2008. So looking at nitrogen mineralization, so we estimated the amount of nitrogen that was possibly mineralized throughout the season from 2004 to 2015. So you can see the green manure, the green line is always the highest. Now, what is interesting is in 2006 and 2007, where we had a drought, you can see that there was some level of immobilization. So the black line across is basically any point below this line is a negative value. So that indicates that when we had low moisture, mineralization was very low. We see that the continuous wheat, which has a high CN ratio, the cereal stubble, mostly immobilization throughout the study, and that basically indicates that a lot of nitrogen fertilizer is actually going to be applied in this system. Similar for fall nitrate and spring nitrate, we do see that there is higher concentrations of nitrate in the up to 24 inches. So that indicates we have lower requirements for nitrogen fertilizer in the green manure system compared to, for example, the continuous wheat system and even the fallow system. I will show you later the differences as we go. So for nitrogen fertilizer applied, as I said, we had low end fertilizer requirements. So green manure was less than fallow, then followed by field pea, wheat, and canola. Now what you can also see is we had a higher nitrogen fertilizer requirements 2 years after fallow, compared to 2 years after green manure. So 2 years after green manure, nitrogen supply was still high compared to fallow. Also, grain yield was higher after fallow than green manure, and lowest in continuous wheat. We had larger yield decline 2 years after fallow compared to green manure. Again, comes back to higher nitrogen supply, as well as on stubble, we did find more of immobilization taking place compared to the green manure. Grain protein was higher in green manure system compared to the other systems, and that basically agrees with the higher nitrogen supply. Now, while the wheat in all the systems received the equivalent amount of nitrogen in terms of both the soil as well as the fertilizer that was applied, for the green manure system, there was a nitrogen credit applied, taking into account 20% of biomass production. Now, we know that nitrogen use efficiency is very low when we have higher rates of nitrogen, and in most systems, a larger proportion of the nitrogen that is actually taken up by a crop typically comes from the soil and not from the fertilizer that is applied. So the ability of the soil to provide that nitrogen is of a benefit compared to applying inorganic fertilizer. Another study I would like to talk about is the Indian head study, and for certain reasons, it's a different growing environment, a different soil type. In this study, it's a heavy clay soil with a higher pH of 7.9, higher organic carbon content. It was established in 1957. It's a no-till system as of 1990. And in this system, the wheat phases are not fertilized. So this would be a good example for an organic cropping system under this context. So we are comparing here the fallow wheat, continuous wheat, and legume green manure wheat. And in this case, it's a lentil green manure. And also compared to swift current, the green manure was terminated with herbicides. So what we found was, we found similar yield after fallow and green manure, but a larger decline 2 years after fallow compared to green manure, which is quite similar to swift current. And also we had higher grain protein after green manure. So that indicates again, in connection with these results at swift current, green manure is able to provide nitrogen even 2 years after compared to fallow. And also fallow is more of a nitrogen mining because there is no actual residue or being applied. So basically just the soil organic matter being mineralized for a full season compared to green manure, where we are adding legume green manure and biomass, which is rich in nitrogen. The last study I want to talk about is a study that is ongoing right now, and where we are using nitrogen 15 isotopes to be able to better understand nitrogen cycling. So we are looking at tracing the fate and flow of nitrogen through the system, both from the crop residues as well as from fertilizer that is applied. So this experiment is taking place in swift current and Lethbridge, designed as a split plot. So we have 6 main plots and 3 nitrogen rates. So our crops are wheat, field pea, lentil, faba bean, and chickpea, and then followed by canola and wheat. Now we also have another system where we have 2 legume phases followed by wheat. So just to explain a bit, so in the first year we grew all of these crops as our main plots, and they're right across the site. Then what we did in the plots that will receive no nitrogen in the subsequent year, so that means when canola and wheat will be grown, once we harvested the crops, we applied, well, at the beginning we applied nitrogen fertilizer that is labeled with N15, that we can get into the plant, and then that gives us the ability to trace the decomposition over time. So after harvest, for some of the microplots, what we did is we returned all of the biomass, excluding the seed. So where it says all, so all of the biomass that was harvested is returned, except the seed exported. In another area where it says BG for below ground, we did not return the biomass. We only left the stubble standing. And that biomass that was in the plots where below ground is, we put it into a new area to be able to trace the decomposition of the above ground biomass by itself. Then in the other plots in the following year, we applied another microplot where we added the equivalent nitrogen rate to be able to look at recovery of nitrogen. And then the following year, when wheat, we repeated the same thing. And the plots that were in the previous year, so the year 2 microplots, they were unfertilized. So we wanted to see what is the residual effect. In the 0N plots, nitrogen fertilizer was not applied, and the same process took place each year. So where we had all, the biomass was returned, BG, only the stubble was left standing, and then that stubble was moved into a new area. So this slide just shows what some of the things that we did. So when we applied our fertilizer by broadcasting, which was more feasible, considering the size of our microplots, so they were covered to capture the fertilizer, ammonium sulfate. Then we labeled the plots with the nitrogen 15 in solution, and then we trapped the residues in these microplots over winter so that they can stay in place and not be blown away by wind. And then once we are going to seed, we remove these cages and seed the next spring. So some of the pictures here, we did have differences in growing conditions. 2019 was a bit drier, especially I think in May, it was very dry. It was extremely dry in July. In 2020, we had about a normal May and June, normal July, and extremely dry August. So we did have some differences in growing conditions over time. So just quickly, so if we look at the blue numbers here, so these numbers are comparing canola seed yield in the unfertilized plots compared to wheat. So in chickpea, we had about 3 bushels per acre more in those plots compared to wheat. However, for faba bean, we had 6 bushels less. In field pea, we had 3 bushels more, and lentil, we had about 1 bushel more canola yield compared to wheat. Now, in terms of the stubble themselves, so in chickpea, nitrogen rate, as you can see for all stubbles, increased yield. We had about 10 bushels more when in chickpea, when we applied about 45 pounds per acre, and compared to 13 when we applied 90. We can see also here that in lentil, applying 50 was lentil and field pea, applying 45 pounds of nitrogen per acre was sufficient to increase yield compared to applying 90 pounds of nitrogen per acre. In wheat, it was 90 pounds per acre was sufficient. Now, interestingly, what you can see here is we have slightly greater soil and supply following legumes, except faba bean, and I will explain why faba bean was not as good. Our yield response to 45 pounds of nitrogen on legume stubble was more than 90 pounds per acre on wheat. And again, those are preliminary results. We are still processing the data. Then we looked at the microplots where we managed the stubble in terms of either having just the stubble standing or all the biomass. So what we found is that below ground N contributed a lot in terms of this canola seed yield. So if you look at chickpea, field pea, stubble especially, you can see here that the below ground was actually higher than where we had all of the above ground biomass. And a lot of studies have suggested that about below ground can contribute about 70% of the total N returned to the soil by certain legumes. We are going to be processing the data more, but those are some preliminary results, but they are really exciting and interesting to be able to better understand what happens in diversified systems with legumes. Where does the nitrogen go, and from where does it, in terms of the contribution, and how can we increase nitrogen credit estimation in these systems. So as I mentioned, faba bean stubble, we did not get good results, and we are still trying to figure out why. The in-crop herbicides that were applied were the same for all of the pulses, legumes, but what we found in the faba bean plots, we had some, it seemed to show that it was herbicide residue, but we're not entirely sure. But what we did see is that when we applied nitrogen, the effect was much lower. So you see in the control plot where we have 0, you can see these huge gaps in canola compared to where we had 90 pounds of N, where the crops were better able to grow. So we are still trying to figure out what actually happened there. So in conclusion, we have found that legumes supply N to subsequent non-legume crops through nitrogen fixation and mineralization of above and below ground residues. The potential and actual N depends on the species, site and environmental conditions, the quality of the biomass, and the yield, as well as whether the legume was grown for seed or green manure. Legumes also provide non-nitrogen benefits, including increasing water availability, reduction in pest and disease cycles, and also growth of beneficial soil microorganisms. Growing in green manure from the study that I mentioned earlier, that Cindy Grant led. So we found growing green manure, field pea or lentil before cereal or oilseed was most likely to increase yields and reduce N requirements compared to faba bean and also chickpea, considering our current study. We did find positive yield benefits in cropping systems that will persist for up to 3 years by the legumes, depending on site conditions on the legume. But importantly, weather conditions, especially in the prairies for moisture, play a critical role in influencing the productivity and then fixation of legume, as well as nitrogen supply, nitrogen mineralization, and the response of subsequent crops. So just to end, just to acknowledge a lot of the funding agencies who have partnered with us and a lot of the collaborators across the country and my research group in Swift Current. I'll be happy to take any questions that you may have.
Thank you very much, Mervin.
Can I stop sharing or do I leave it?
I think you should keep sharing because we might want to go to some of the slides. But so I'm assuming you can hear me based on your question.
Yes, I can hear you.
Okay, great. Well, thank you very much. We are going to open it up for questions. And while the questions are coming in, I did want to make some observations. One of them is that you have provided some very good quality research. Some of it comes from conventional production, but I think it is really important for the agronomists on the call and also the farmers to be able to discern useful information for organic production from conventional research, because there is such great research going on, and you've demonstrated that, especially these long-term studies, they are amazing. And so I just want to challenge our listeners to continually look for nuggets of knowledge which they can apply to their organic system from the type of studies that you are engaged in. Let me just check on the questions to see if we have any yet. Yeah, we don't have any questions coming in yet. I got Janine Gibson saying that her screen was black. I'm sorry about that. While we're waiting for some questions to come in, and if there are questions that I'm not seeing and our webmaster is seeing them, then please just unmute. Just let me know, please. I thought your observation, Mervin, in 2006, 2007, you said you showed that there was a drought, and so that year you had very little mineralization of nitrogen. So what is your recommendation to organic farmers who would be in a situation where they have produced quite a bit of green manure biomass, but it was dry, like in 2006, 2007? Am I correct in assuming that they're not going to get a lot of nitrogen from that green manure biomass? Is that a correct assumption based on your work?
So yes, and when I say that it was dry, it's not the year that the green manure was grown. I'm talking about the year subsequent, the subsequent year.
Correct.
So
right.
The moisture that is required to activate microbial because it has to be soluble, okay? And the microorganisms need to be able to move around. So without the moisture, they can't move around. They can't attack the residues that are there. So actually the residue would almost remain not totally intact, but it will not be decomposed as you would have expected in a normal adequate moisture environment. So the nitrogen availability will definitely be lower. It's the same as when we, in a conventional system where we apply nitrogen and then there is a drought. Most of the nitrogen is going to be in the soil. It's not going to be taken up by that crop. So having a soil test at the end of the season, you will find elevated levels of nitrate. And you will think, well, your crop, that year, the next year you're going to apply the same amount of nitrogen, but the nitrogen that you applied the previous year, a lot of it is still tied up in the soil. So you have these high levels of soil test N. So it's the same scenario with crop residues.
Yeah. So what can a farmer then assume if we had good green manure growth in 2020? In 2021, there was no rain to mineralize it. Will we get that nitrogen in 2022 if we have moisture? And how much of it is still there?
Exactly. It will be eventually mineralized when the conditions become suitable. As I showed here, 3 years after, we did still see huge impacts of legumes that were grown 3 years prior. So there is still mineralization taking place. In the nitrogen 15 isotope experiment that I am conducting, that was one of the justifications behind that experimental design, where we have in the control, we are trying to see how long are those residues going to be providing nitrogen. Can we still detect, because it has the isotope, so we can differentiate between the nitrogen 15 and the nitrogen 14, which is normal nitrogen. So we will be able to see how long this will be able to be mineralized and supply and detect it in the crops that are being grown in those areas. So yes, a farmer should expect mineralization once conditions become suitable. Okay.
That's great. I'm okay. I did get a question here. By the way, just that's really interesting. And what I also found really important about your presentation is that it showed the value of growing a legume green manure instead of just fallowing the field.
Right.
And if we know from short-term experiments, sometimes it doesn't matter, especially if you have high organic matter soils. But I think one of your gifts to the audience today was to really demonstrate and drive home the fact that growing a green manure regularly in the organic rotation is going to be important to the long-term sustainability of that system.
Exactly. The green manure is, look at it as your bank. It is a source. It replaces inorganic fertilizer. So having a fallow, as I mentioned, is basically you are mining the soil organic matter. You're exposing the soil to all of the conditions, so wind erosion and so on. And it's mining without replacement.
Yeah.
The green manure will replace. It adds to our soil organic matter. And one of the interesting things about soil mining is, or priming, that can come into play here. So when you add a residue, depending on the CN ratio or the quality, you can have two different types of mining from theory. There is one called co-metabolism, where if you add a fresh organic matter or fresh organic residue, that serves as an energy source for microbes. And then they can help to degrade soil organic matter, as well as degrade the residue that you have added. So it enables mineralization. Then there is the end mining. So in the end mining is where you have fresh organic material added, like green manure. Now that organic material will stimulate microbial growth. Now, in a situation where you have nutrient limitation, especially nitrogen, you will find that the microbes will then mine the soil, not the residue, to get that nitrogen to decompose that residue that you added. So in that case, you have nitrogen mining from your soil, not the residue.
That's really fascinating. I think what we need is for some, that is just so important. We need some very clear illustration of that. But when I think about that, I think of little Pac-Mans, and you know, they can either eat the nitrogen from the plant residue or they eat the nitrogen from the organic matter, and that's a really interesting distinction.
Yeah, and the end mining will likely be more apparent in a higher CN ratio residue, so like a cereal, because it has more carbon, less N. So to get the amount of nitrogen to decompose that cereal, it has to find the nitrogen somewhere else. So that you will get it from the soil organic matter.
So if we don't put a green manure into the rotation regularly in an organic farming system, and we rely on tilled fallow, then we are, because of the mechanism that you explained, we are going to be mining the organic matter and reducing the carbon in the soil, correct?
Exactly, over time. So we have shown in the fallow systems, soil organic carbon is actually decreasing.
Yeah.
And in the green manure system, it fluctuates, but it's not on a downward trend compared to the fallow. Okay.
So for the organic farmers who are part of, Farmers for Climate Solutions, the new organization, adding those green manures or cover crops, if they're maybe in Eastern Canada, is really important to maintain their soil carbon or grow their soil carbon. Yeah,
exactly. Carbon, soil organic matter, it is plant carbon. So the more that you add is better. And there is, basically it is the sort of the difference. It is a balance between addition and output. So the output could be decomposition, loss of CO2, whatever. And then the addition is either a green manure crop is grown, you have biomass return and all of that residues to be able to balance off. Because in swift current, especially, When we have dry conditions, we have increases in soil carbon. Whenever we get, and that's from the long-term experiment, whenever we see that there is optimum conditions, even with good growth, it might not be good enough to balance off the losses because the conditions are so good, the decomposition is fast. So we have to produce more biomass and more plant carbon to exceed the losses to be able to maintain or increase, even in a semi-arid.
Okay, so for an organic farmer then, Mervin, I know there are a couple of questions that came in. I'm going to get to them right away. But so for an organic farmer, when like this year in Manitoba, where the rains started coming in some places in early September, are you saying it would have been better for the soil carbon accumulation to actually plant something in that field to add carbon than just to leave it fallow?
Well, I would always suggest planting something in the field compared to fallow.
Okay.
Because fallow, yes, fallow actually is, well, I mean, was designed to conserve moisture. That was the purpose for fallow. And nitrogen mineralization is a side benefit of fallow, but that's not the true, at least from my understanding, not the true reasoning behind fallow. It was for moisture conservation. So leaving a field fallow is not the best option unless we have, I would say, consecutive years and years of drought that we can't have a crop. That would be the only scenario I would recommend growing fallow. I mean, having fallow compared to growing a crop, even if it's just a cover crop.
Okay. I'm going to just ask you one other thing here. You're getting grilled. You didn't think he's such a grilling here, but there are a couple of questions here, but I want to ask you one last thing before I get to the questions. You made an interesting point, I thought, in that you said that under dry conditions, terminating your green manure at 20% flower was better than waiting till it went to full flower. And in a, Manitoba is traditionally wetter than Swift Current, but how much more, so maybe, but in dry years, people think about this. So can you expand on that a little bit? How much water did you save by terminating the crop at 20% flower versus full flower?
Yeah, that's a very good question. I do not have the actual data of in terms of the moisture that was conserved, but just the when I looked at the long-term data set, what I saw was, and some of this has been published already, especially from Bob Zettner. There was lower soil moisture after when the green manure was terminated at full bloom compared to when it was 20%. So when they started terminating it earlier, they had more moisture in the soil. The wheat after green manure, the yields were better compared to when they terminated it at full bloom. And so the decision was taken to terminate earlier. And because as long as you have the crop growing, especially late in the season, August, it's gonna use water. And then if we do not have rainfall, if we have low snowpack, then we are not able to fully replenish the soil. So you want, in a green manure system, you want biomass, but at a certain point, the crop is not going to produce more biomass. It is in a reproductive stage. So once you go to flowering, you are not really getting more biomass, and you are actually increasing carbon levels compared to N. N will then be depleted in the plant because it is now transferring all of that N to the seed, to seed production. But you want the N. You do not want the crop to transfer the N to the seed. As I showed, a huge portion of the N is exported in seed. So the earlier, I mean, once the crop reaches maximum biomass, I think this is the ideal time. You do not want the crop to produce pods and so on. That means unless you are putting the pods back into the ground, but you have to also look at it has also used water to do that.
Okay.
So I think it's best to terminate earlier compared to later in a dry environment for moisture conservation.
Thank you for that. That's really helpful. Okay, I'm going to get to my two questions here, and please send more questions. So the first question was that 70% of nitrogen that is below ground of the total plant end, does that include or exclude the seed?
But that's excluding the seed. That's
Okay.
not the so the seed is not considered. I'm talking about the stubble, so the roots basically. A lot of the pulses, legumes, they got, remember, this is where the fixation takes place, is in the roots. So that's where all that mutual relationship is. So it is quite expected that you have that higher nitrogen concentration in that area. And remember that the fixation also is in the soil.
Yeah.
So
what about Mervin? So then what I understand is that's a situation where you would grow like a pea or a lentil crop and you would harvest the grain, and then what's left over, 70% is in the soil. What about in a green manure situation where you grow that crop just to incorporate it into the soil? Where is the nitrogen in those situations? How do the percentages line up?
So that's a good question. Now, just to explain a little bit better, now that 70% is, let's take the plant, the legume. We have a lot of the nitrogen exported as seed. But if we take the root only and analyze that, we will find in some cases even equivalent with the seed in some cases. For green manure, the entire biomass, including the roots, so you have your full 100%, okay, because you do not have any seed exported. I do not have actual values to differentiate in a green manure what proportion is in the seed, in the root compared to the above ground. But considering these data sets that have been published, even in Australia, they did find about 50 to 60% of the nitrogen in the legume crop. It did not specify whether it was a green manure or grown for seed. Most of that was below ground. So considering just a green manure, most of it will still be below ground, not above ground. So in that experiment that we are doing right now, clearly taking off the above ground biomass, we still had yields that were equivalent to where we had everything, even higher in some cases. So this is not to encourage removal of biomass. This was just to demonstrate whether or not the stubble itself without the biomass can still contribute. It's not a practice that we want to encourage, but just for experimental purposes, we designed it like that. So it's clear that the stubble is the big contributor.
Yeah.
Even if look at the lentil, it's not a big crop. You barely have any stubble in the field after you harvest, but you still find it is providing enough N. The field pea did provide more than lentil because it is the bigger plant compared to lentil, and chickpea is a little bit bigger than lentil as well. But overall, the stubble is a big contributor in terms of nitrogen supply.
Okay, thanks. That's really interesting. I'm going to get you. I'm going to ask you about mixtures in a minute, but I'm going to go to my next question here. In the mineralization of nitrogen and the nitrogen uptake, and maybe you got to this already. If it is available, the person is wondering is kind of the question that I asked. If it's a dry year, is it available in future years? And do you think the nitrogen volatilization differs depending if the nitrogen is available in year 1 or in year 2? So the question was, how much do nitrogen, how much does, are there any losses due to volatilization that people might need to be learning a bit more about?
Yes, that's a very good question. So volatilization typically occurs when we broadcast. Okay,
Okay, we're not talking about fertilizer here. Just to keep you.
I know, but even using the biomass on the surface. So let's say in an organic system where we do not have the residue being tilled into the soil and it's left on the surface.
Oh, okay. Okay.
There will be some volatilization when conditions become suitable. Having that residue buried or somewhat buried will reduce that the proportion of losses. Now, the nitrogen that is in the system when it's dry likely stays there. With any moisture, you get a burst, you get a flush, you get an instant activation of activity, and that can contribute to losses in different ways. So you can still have denitrification, especially if it's extremely wet. You will have some, depending on how much precipitation or the soil type, it's possible to have some leaching. Leaching is not a huge phenomenon in semi-arid, mostly in the humid conditions, but we have to keep in mind, nitrogen that is not taken up by crop or it is in the soil is not entirely lost in the system. It is just in an unavailable form at that point, or the crop is not able to access it because the crop is not growing. Or if it's a residue in a green manure system, it is not mineralized, it is not decomposed. It is in its form that, so basically you terminated the crop is still there. So yes, it can become available 2 years later, as we showed for the long-term experiment, and it will not be under normal conditions. The availability will be higher as the year after, and then tends to decline as we go further away.
Okay, and at the beginning, you did say a little bit of soil incorporation might be helpful. Yes, because actually you have more contact with, you have breaking down of the residues themselves, and that's what I think that was why in the new rotation they implemented that system to have a shallow tillage. However, I will mention there was a study in 2016 that was done in Swift Current. I was not part of that, but they compared green manure and other pulses to, lentil, green manure and some other pulses to different methods of termination. So they had herbicide termination, they had tillage, and they had frost. And what they found was, if I remember correctly, there's a paper from Patrick Mulecki. That's in the Canadian Journal of Plant Science. So what they found was, among those different termination methods, there were no differences in moisture in fall, no differences in moisture in spring, no differences in nitrogen mineralized and nitrogen uptake in the subsequent year. That was just in Swift Current, okay? But that study did compare those methods. So that's a paper that you can take a read. That's really interesting. And when we started working with the blade roller and we left our green manure on the surface, we did definitely notice losses of nitrogen. We got more nitrogen out of it if we incorporated it. So I'm going to get the last question, and I'm going to give you about 2 minutes to answer it. And then we're going to wrap up here. So, okay, in the N15 study, why was there little benefit from the legume when there was 0 N applied.
Compared to?
Do you know what level of soil moisture is required to mineralize nitrogen?
That's a very difficult question to answer in terms of what level of moisture under controlled conditions. So let's say we are doing an incubation, we do adjust our moisture to about 60% of the soil's water holding capacity, or say 60% water field pore space, somewhere around that line. That's optimum. In a field condition, what is the exact number? I at this point, I am not able to answer fully. I would have to look into that. But definitely having any moisture is going to enhance mineralization. Once, if you have your crop that is growing well, good yields, that means there's also mineralization taking place. If the crop is always a good indicator of soil conditions. So if your crop is struggling, then likely you do not have the expected mineralization taking place. Now, in terms of why did the crops in the pulse plots that were not fertilized do poorly? Well, I wouldn't say they did poorly. They remember, in those plots, the crops are only depending on nitrogen from mineralization. So there was no N added. Compared to the other plots where we had 45 pounds and 90, we do have more N added. So obviously, in Swift Current, which is a very low soil organic matter content soil, so the crop is going to depend almost entirely on that residue or on that pulse. That was the previous year. So we do expect higher yields when we apply nitrogen, even in a pulse phase. But the big question is, how much nitrogen do you need to add?
Yeah. Another question came in here quickly, and I'll just somebody asked about why did faba bean do so poorly, and I know you talked about the herbicide residue as a possible reason. What's your feeling? Would you recommend faba bean as a green manure?
Yes, as a green manure, as I showed in the other studies across many sites in Western Canada, it did perform the best. It did perform the best. But in our case, we are not entirely sure. As I said, all of the pulses received the same herbicide. Only in the faba bean we had that impact. So we are still looking at our notes and our data to find out what possibly happened there. We are not sure at this point. Okay,
because it is such a good nitrogen fixer. So Mervin,
yes.
Out of time. We are out of time. I feel like, Rosalie Barton here, or Rosemary Barton, cutting people off, CBC, but I've had a ball. I've had so much fun learning from you. And I think the farmers have as well, and the agronomists on the call. So we're going to turn it over to Karen now, but Mervin, thank you very much. And we will.
It was my pleasure.
And I'm sure farmers will have a chance to learn from you in the future. Take care. Over to you, Karen.
Great, thank you so much. And yeah, thank you, Mervin, for your presentation, and Martin for facilitating. I see there are more questions in the question box that we did not get to. We're gonna email Mervin those questions and ask you if you'd be able to respond via email, if that'd be all right.
Sure, no problem. I will answer them as best as I can and as quickly as I can. It was a pleasure being here.
Great, thanks so much. Yes, so lots of interest and attention, and really relevant for the year to come as we plan what our next crops and how we should be managing them. Great. So I just wanted to thank again our sponsors and our funders. We've been receiving we have a grant from through the Canadian Agricultural Partnership that we received through the the Prairie Organic Development Fund as well. We also have been receiving in-kind support from Manitoba Agriculture through the time of Laura Telford and Lori, who's been facilitating this webinar as well. So thank you guys very much for all of your contributions. We would like to thank FCC, who's one of our sponsors. We have Grain Millers, who's sponsoring us as well. So if you are interested in an oats or flax selling or contracts for next year, give them a shout. We also have sponsorship from Manitoba Harvest, and if you're interested in growing organic hemp, I think they've got some contracts out and ready for next year as well. So give them a shout. We want to thank ProCert for also being a sponsor of this webinar, and Theory Mesh. All right, so thank you so much for attending this webinar. We'll be back again next week on November 30th for the next in our series. So thank you so much, and we'll see you next time.